Okay, so chapter 6 is all about the cell membrane. Chapter 7. The chapter 6 is all about the different organelles and so on. And while one of the kind of structures is the cell membrane or plasma membrane, there's enough to it to devote to a whole chapter to it. So it's all about the membrane, both its structure and its function. And we know a little bit about the structure. So the overview, the plasma membrane is the boundary that separates the living cell from the non-living surroundings. So, and remember, it controls what goes in and out of the cell. So the plasma membrane exhibits what we call selective permeability. So it's selective permeability based upon certain characteristics about molecules and so on that we'll learn about. So it picks and chooses what can go across it. So it allows some substances to go across more easily than others. And so this is an overview picture that we're going to look at here of what the cell membrane is composed of. So basically, here's your lipid bilayer, and we'll go through these as we go along. We'll talk about each one of these. These are proteins embedded in the membrane, so that some proteins go through the membrane, some do not. Some are just on the interior, and the proteins go all the way through. So if you're looking at here, these are little proteins sticking out, but you would see this through it like you do here. Inside here, there are 4 fused rings right here. I don't know if you can see them right here. 4 fused rings. What do we know about those? Those are steroids, and so this is cholesterol in the membrane. And then we have these little carbohydrate chains on the protein. And then this part right here we'll talk about in a little bit here. It's called the extracellular matrix. Actually, let me talk about that real quickly right now. There's these proteins. Some are fibrous and some are long, these like worm-like structures that connect. Notice here, these things connect to one another. And the purpose is this is cell 1. Let's call this cell 1. You could have the membrane of another cell right here, and you have these proteins in the membrane here connected to these parts of the extracellular matrix. So these guys here, this is the extracellular matrix, and it kind of connects cells. So they're connected from cell membrane to cell membrane. So this is cell 2 right here. This is the membrane of cell 2. So that's what that picture is here. And this is the cytoplasm inside this cell. Okay, so let's look at the cell membranes are fluid mosaics of lipids and proteins. So fluid mosaic, fluid, things move around. So it says blank are the most abundant lipid in the plasma membrane. You should know this. What is it? What kind of lipid makes up the plasma membrane? It is the phospholipids. Most abundant. They are, it says blank molecules, meaning that they contain hydrophobic and hydrophilic regions. So we call that, they're called amphipathic molecules. So if you recall, when we looked at the structure of the phospholipid, that we looked at them in terms of drawn like this, where they have the head region and the 2 tails coming out. But remember that really the structure of it, you have that 3-carbon glycerol molecule connected to 2 fatty acids. So these are fatty acid chains. And instead of a 3rd fatty acid, you have a phosphate group. And so this part right here, the phosphate group here, is called the head portion. And that, remember, is polar or nonpolar? Polar, right? And then you have here the fatty acids, the tail region, and these are nonpolar. They're hydrocarbon chains. And so in the lipid bilayer, you have this, so that the heads that are polar are facing towards the water inside and outside of the cell, and the tails are in the center. So the fluid mosaic model states that a membrane is a fluid structure with a mosaic of various proteins embedded in it. So you have these proteins studded around, and you have these lipids, and it's fluid-like. They say that the membrane has kind of the consistency of salad oil, so it's pretty fluid. So let's look at more directly this fluid mosaic model. So membranes have been chemically analyzed and found to be made up of proteins and lipids. Scientists studying the plasma membrane reasoned that it must be a phospholipid bilayer. We just talked about that. In 1972, two scientists by the last name Singer and Nicholson proposed that the membrane is a mosaic of proteins dispersed and individually inserted into the phospholipid bilayer. So you can see that they're dispersed around here, sticking out of that phospholipid bilayer. Let's turn it over here. So this picture here shows you the bilayer, all right, and which was what I was trying to show earlier. So this is showing you the phospholipids, and so these guys here are the tails, right? So this is nonpolar tails on the inside, and this part right here, these are your polar heads. And you can see right here, trying to show you the molecules and atoms there. These are waters, all right, and so therefore they're attracted by the opposite charges here and here, but not attracted to the hydrocarbon. And so that's what we have there. So in 1935, Davison and Danili proposed kind of a sandwich model in which the phospholipid bilayer lies between two layers of globular proteins. Globular proteins, so like this, all right? They're not like the thread-like proteins. All right, they are kind of a blob. And so later studies found problems with this model, particularly the placement of membrane proteins, which have hydrophilic and hydrophobic regions. So the membrane proteins have hydrophilic and hydrophobic regions, which means they're amphipathic, that whole word here containing hydrophobic and hydrophilic regions. So in 1972, Singer and Nicholson proposed that the membrane is a mosaic of proteins dispersed within the bilayer with only the hydro what regions exposed to water? Hydrophilic, right? And that's what they actually found out. All right, so the proteins aren't like in between sandwiched in between the phospholipid bilayers. The proteins are actually embedded and go through the whole thing. And they actually have hydrophobic parts and hydrophilic parts. So here, the protein, this is the hydrophobic parts of the two tails of the phospholipids. All right, we know those are hydrophobic there. But also, the protein has the hydrophobic parts. All right, so this is also hydrophobic. And so all in orange is hydrophobic. And then the hydrophilic parts are the parts exposed near the water. All right, and all the heads are hydrophilic as well. All right, so therefore proteins and phospholipids are amphipathic in the lipid bilayer. So to further their study about the cell membrane structure, freeze-fracture studies of the plasma membrane supported this fluid mosaic model, that these proteins are kind of scattered around this lipid bilayer. So what they did is a specialized preparation technique that splits a membrane along the middle of the phospholipid bilayer. So it's like they freeze it, and see how there's nice two layers here, and they kind of chisel the two layers apart, so it breaks it into two layers, and kind of saw what was in the middle. And this is what they saw. So here's them freezing. This is the cell, so the plasma membrane's frozen, and I'm using microscopic little knife here, kind of broke apart the plasma membrane, the lipid bilayer. And what they found is that when they separated it, the proteins were sticking out of this layer, and there was holes in the membrane here. And so therefore that proved that proteins actually went through the entire layer, and it wasn't like a sandwich of lipids and proteins. And so that proved their theory about where the proteins were at and how they were scattered through there. So then the fluid part of it, let's talk about the fluidity of membranes. Phospholipids in the plasma membrane can move within the bilayer. Most of the lipids and proteins drift what's called laterally. All right, so what does this mean? So within the bilayer, so if you look at here, this is lateral movement, meaning it stays in the same layer. All right, so in same layer. So notice here about 10 to the 7th times per second. So these guys this is this arrow here going like this, showing you that they can move. So they're not, stationary in one spot. This lipid can eventually move and be over here as time goes on. But they usually stay within the same layer. Notice that it's a very rare occurrence. This arrows are supposed to be showing you like this. Very rare occurrence where they flip-flop layers. So about once per month here. So the fluidity, remember fluids are due to the kinetic energy of the molecules. And so therefore this movement, it means that it's fluid, all right? Because that's what happens in liquids or fluids. So what happens as temperatures cool, so it's cool, what happens to the movement of the phospholipids? The movement goes down, right? The kinetic energy. And so that's how something goes from a fluid, a liquid, to a solid, right? It freezes. And so as the temperature cools, membranes switch from a fluid state to a solid state. And that is not good for a cell. If the cell freezes, if it freezes solid, things aren't going to be able to go into and out of the cell, and so you're not going to be able to get things in and waste out. And so the temperature at which a membrane solidifies depends on the type of lipids that are in it. So do you guys remember what kind of phospholipids, what needs to be true about them for them to maintain some spacing in between them? Remember, there were two kinds of phospholipids, ones that the fatty acids were straight and ones that had the kinks in them. And remember, the kinks don't allow adjacent phospholipid molecules to get that close to one another, so therefore maintain space. So if the temperature was getting cool, which kind of phospholipid would be best for the cell membrane to be made out of? It'd be this one, right? It would take longer for this one to freeze because of that spacing. So are these saturated or unsaturated phospholipids? Well, they have a double bond, which means they are unsaturated. So membranes rich in unsaturated fatty acids are more fluid than those rich and saturated, and can maintain their fluidity longer, especially in cooler temperatures. And so membranes must be fluid to work properly, so they are usually about as fluid as salad oil, as I said. And different cells will have different concentrations of different types of lipids. And we're going to see that some organisms can actually change, some plants can actually, as winter comes, they can actually change and make it so that their phospholipids have more double bonds, and so therefore become saturated and therefore have a resistance to freezing. This is kind of a little interesting tidbit there. So that's my picture. The book, of course, has a much better picture. So here's fluid, unsaturated hydrocarbons. So that means each one of these kinks, there's a double bond. These are saturated. So this more viscous, thicker, because they're spaced closer together. One other type of lipid that's found in the membrane is a steroid. And do you remember what the steroid is called that's found in the membrane? Cholesterol. The steroid cholesterol has different effects on membrane fluidity at different temperatures. At warm temperatures, so 37 degrees Celsius, cholesterol restrains movement of phospholipids. The 37 degrees is about the human body temperature, just to give you an idea of, it's about 98 degrees. And so the cholesterol, here's cholesterol. Let's draw this in here. Let me so here are your cholesterol molecules. Let's draw those in so you can see where they're at. And so they're embedded in between these. So at warm temperatures, it restrains movement of phospholipids. So these are in between the phospholipids. So remember, the phospholipids can move within a bilayer. And so it makes it hard for them to move around because they're kind of blocking their way and so on. So it restrains movement of the phospholipids at a warm temperature. But let's look at cool temperatures. So it's the opposite. At cool temperatures, it maintains fluidity by preventing tight packing. So it makes it so that it doesn't get too fluid, too much movement in warm temperatures. So let's put here, reduces fluidity so we can compare. But so it makes it from being too liquidy, and then here it helps to maintain its fluidity when the temperature goes down by preventing them from getting too close together and solidifying. So therefore, either way, warm or cool temperatures, the cholesterol helps to maintain the correct fluidity for the cell membrane. All right, so cholesterol is very, very important. Another big important thing is the proteins. So let's talk about those embedded proteins. Some proteins in the membrane can drift within the bilayers. They move around. Proteins are much larger than lipids and therefore move much more slowly because a much bigger molecule. So they've done some investigations about the movement of proteins. So to investigate whether membrane proteins move, they fused a mouse cell and a human cell. So let's see what happens in this experiment. I'm going to get a couple of colors here. So here's your mouse cell, and these are the membrane proteins that are sticking out. So I'm going to color code the mouse cells pink here real quick. Let's put a dot on each one of them. And then the human cell is green. So we have the green proteins. Just put a green dot on each one of these. All right. So then what happens when they fuse? So they fuse them together, so the membranes fuse together. So we still have all the green ones here. So this is a hybrid cell, a mixture between humans and the mouse cell. And then what do you think happened over time? What they found is, so you have the one big membrane here. They found after only an hour, not a very long period of time, that the human and, so I'm going to put some human proteins here, they were intermixed. And so here's the mouse. All right, so they all moved around, and you found some in each, meaning that are each area here. So they moved around, and let me put a few more. I didn't color in all of them, but you get the idea that they moved around, and this proved that the proteins are not static, that they move. So let's write this here, proved proteins move through out the bilayer, the membrane, I'll just say. So they don't stick in one spot. So these proteins are a big part of the membrane. So let's talk about their functions. So a membrane is a collage of different proteins embedded in a fluid matrix of the lipid bilayer. Proteins determine most of the membrane's specific functions. They have a big role. So there's two different kinds. There's peripheral. Think about the word periphery. If you go around the periphery of like a field, you're going around the outskirts around it. So these are not embedded within the protein, but they are on the outskirts of it. And then integral proteins are the ones that go through the membrane. So we see here integral proteins. Let's get a different color here. Go all the way through the membrane and have that, remember, hydrophobic and hydrophilic region, integral proteins. So all of these would be integral proteins. And so I'm just going to go ahead and do that real quick. All right, for integral proteins. And then we have our peripheral proteins. These are just on the inside. So here we go, peripheral proteins. We can see right here, right here, right here, and right here. Okay? And so those are your two different kinds. So let's talk about the integral proteins. Integral proteins that span the membrane are called, and we're going to call those, let me get my pen here, transmembrane proteins. They go through the membrane. The hydrophobic regions of these types of proteins consist of one or more structures of nonpolar amino acids. That's what makes them hydrophobic, all right? So remember that there were amino acids that have the polar R group. There's the, so this is talking about the R group, the electrically charged or nonpolar. And so what we're looking at here then is, remember that, let me get a different color here, let's do purple, that this protein here is really a chain of amino acids. All right, so this is a polypeptide amino acid chain, which means that if this part right here is in the hydrophobic part, the amino acids here have nonpolar R groups. And therefore, that's what makes them, this part, nonpolar. So then we can conclude that this part and this part are polar R groups, right? And so that's what that means. So those are your integral proteins. Let's turn the page here. So that's what this picture is showing you here. So you have this picture, well, let's look at just this one down here. This blob in the membrane is a protein, but realistically it is a chain of amino acids. So here's your big protein embedded in the membrane, but that protein is made out of a chain of amino acids. And so that's what this is trying to show you. And so this would be right here, your hydrophobic. And these guys, hydrophilic region. So what are these functions of the membrane? They can be involved in transport, enzymatic activity, signal transduction, cell-to-cell recognition, intracellular joining, and attachment to the cytoplasm and the extracellular matrix. So let's talk about each one of these here. So here's transport. So some of those, as I talked about before in the chapter's, last chapter's notes, that some of these proteins have a tunnel through them that allow molecules to go into and out of the cell. Some actually carry them. The protein actually changes shape, so you can imagine that this opens up. So this side closes and this side opens up and allows molecules to go through. So that's both involved in transport. Sometimes enzymes aren't free floating in the cell. Sometimes they're actually embedded in the membrane. So remember, enzymes speed up chemical reactions, and so some of those enzymes can be a part of a cell membrane. Sometimes they're receptor molecules. They're involved in receiving signals from another cell. So an example of a signal is hormones. Hormones are molecules that are released from one part of your body and go to another part of your body. And so when you look at a signal molecule, this comes from another cell, and this signal molecule comes to this other part of your body, binds to this receptor. Notice the receptor has a complementary shape, binds to that receptor, and causes a change to happen. So it communicates with the cell inside of here. So causes a change to happen inside the cell. All right, so this is how cells communicate with one another, one cell to another, because they can't talk to one another, but they can communicate through molecules. Cell-to-cell recognition is next. So cells that are adjacent to each other can bond together and therefore recognize one another. And let's actually talk about that down here. Cells recognize each other by binding to surface molecules, often carbohydrates, on the plasma membrane. Membrane carbohydrates may be covalently bonded to lipids, forming what are called glycolipids. So glyco in front of something means it refers to carbohydrates. So if it's a lipid that has a carbohydrate attached, it's a glycolipid, or more commonly, the carbohydrate can be attached to proteins forming, what do you think we call those? Glycolipids. So carbohydrates on the outside, the external side of the plasma membrane, vary among species, individuals, and even cell types in an individual. And I talked a little bit about this before, but let's now relate what we just wrote here back up to this picture here. So here is a glycoprotein. It has this carbohydrate chain. So this right here is the carbohydrate chain. And this kind of, I like to think of it acting as an ID tag. So it's cells recognize one another. So a cell of the same individual will have certain carbohydrate tags, and they have also receptor proteins that can recognize those ID tags and say, oh, this cell belongs to us. So that's why it says it varies among individuals, and also among species, so from one type of organism to another, and even sometimes cell types, the types of cells in an individual person, in an individual body, different cell types will have different carbohydrates attached. So it's a way for other cells to tell, does this other cell belong to us or not? Sometimes it's intracellular joining, so that's so again cell to cell, and that's a way that they kind of stick together. So they join together, kind of like bonding together, two proteins. And then lastly, attachment to the cytoskeleton inside the cell. So here's the cytoskeleton. This is those microfilaments there, and the integral protein right here is bonding to that, or to the extracellular matrix on the outside of the cell. So that's supposed to be showing you that here. That's how it binds to that. And so this is extracellular matrix and the cytoskeleton. So let's talk about membranes just in general, how they're made and sidedness of them. So membranes have distinct inside and outside faces. This asymmetrical distribution of proteins, lipids, and carbohydrates, the three major parts of the cell membrane, is determined when the membrane is built by the rough ER and the Golgi apparatus. So remember back with the endomembrane system, and how the transport vesicles fuse with the cell membrane and so on and help to build membrane. So let's turn this over and see what I mean here. So this is kind of a review of the endomembrane system, but putting something else a little bit into it as well. So if you recall, here's your rough ER, and that's where proteins are made. But the membrane of the rough ER, if we were to look at the membrane, if we just blow that up a little bit, is a lipid bilayer, and it also has proteins embedded in it. So it's very similar to the construction of the cell membrane. And so what this means here is that there are proteins, that's what these guys are, I'm going to put in purple here, embedded in the membrane of the ER. And so what happens is at the ER, they put carbohydrates on these proteins. That's why it says transmembrane glycoproteins. So I'm going to put here an orange here. It adds a carbohydrate to the protein. And so then remember that transport vesicles butt off and go to the Golgi apparatus. So it makes its proteins. I wanted to make sure that the proteins that it's making to, let's say, be secreted from the cell. So these guys are going to be secreted from the cell. It's making those proteins here. They're different than the proteins that are embedded in this membrane. And so what happens is now we get our transport vesicle, and this is your embedded protein with the carbohydrate attached to it. So I'm just going to put a little orange dot here. And it still contains the protein that it made here. This transport vesicle fuses with the Golgi apparatus. And so when it does that, now that transmembrane protein is part of the membrane of the Golgi apparatus. And so I'm going to draw here the carbohydrate attached to it. And then throughout the Golgi apparatus, an additional that the Golgi apparatus does is now you have these glycoproteins as part of the membrane, but you also have the Golgi apparatus making glycolipids. So those lipids here, one of the things that the Golgi apparatus does on the inside here is to add carbohydrate chains. So that's what this here is a glycolipid. They're adding carbohydrate chains to the lipids so that when the vesicles bud off of the Golgi apparatus, you not only have the purple proteins that were once part of the ER with the glycolipids proteins with the orange carbohydrate, but you also have the glycolipids as well. So you have the glycolipids on the inside. And so what happens is when this fuses with the membrane, what happens is all of these glycoproteins and lipids that are sticking out on the inside, when it fuses, it's going to be on the outside here. So here's the glycolipid, and here's the glycoprotein with the carbohydrate attached. So now when it fuses together, can you see that the glycolipids are on the outside now? So it's actually part of the membrane, and the glycoproteins are on the outside, and it's the carbohydrate chains that are hanging off here, like that. And so those are involved in like cell-to-cell recognition, these carbohydrate chains and so on. And so the fact that they are made and put on the inside of all these, that's the purpose, is so that when it fuses, that it is on the right side of the cell membrane. And so that's your plasma membrane. And so then it still secretes the protein that it originally made, but it also puts new glycoproteins and lipids in the membrane. And this is where I would like to stop.